Hello. I think we can get started. You know, we have this tradition at Fox Chase of doing what we call distinguished lectures, and generally they're outside speakers, and generally they're on a Thursday. Well, I think today is Wednesday, and the speaker is an inside speaker, yet it's hard to say that, you know, a seminar by Baruch Blumberg is not a distinguished lecture. It certainly is. So we're going to make an exception today. It is a distinguished lectureship. At least I'm decreeing it so. You know, Barry hardly needs an introduction, either here or anyplace else. Nevertheless, I did find from just talking to him now that he hasn't given a scientific talk here in a number of years, so it's a particular pleasure to get him to talk about some of the historic developments that led to the discovery of hepatitis B and also more recent things in that field. You know, I don't have Barry's CV here, but I do remember there's a couple of interesting things on it, like Nobel Prize. That's pretty good. I think we win that, but the other ones hardly make any difference, but he, of course, won many other awards as well. I also recall a couple of years ago, I was doing an analysis of Fox Chase authors and their impact, you know, citation impacts, and I don't recall the exact number, but it was phenomenally high. It was definitely the highest that of anyone at Fox Chase. You know, just shows that a lot of people have read his works, a lot of people have cited his works, and that there are many such works. You know, I could go on, but I'm very pleased to have you here, Barry, to talk about some of the work at hepatitis. And I'll just close by saying that, you know, as important as the discovery of hepatitis B was in infectious disease and cancer control, etc., in some ways, and I get this from reading your book, I think the method behind it was more important than the discovery itself, because that method applies in a broader way to other discoveries that are still being made now, and in fact maybe being made at an accelerated rate with the new tools we have was not available at the time you were most active. So great pleasure to have you here, Barry. Thanks.
Thanks, John, for that favorably brief introduction. It's a pleasure for me to talk to you. By the way, there are seats down front, which I think have some people standing back want to sit down. I think I probably haven't spoken about hepatitis B at this institution for at least 30 years. I gave a talk on my 80th birthday, but that was about space and about astrobiology. And actually, I'd like to come back and talk about that, but I thought it would be remiss to have my first talk in 30 years about a subject that didn't deal with cancer. I'm glad that John referred to the method that we used in this discovery, because it was, in retrospect, a kind of a unique organization we were able to have here at Fox Chase. To begin with, when I came, Dr. Timothy Talbot, who is the second director here, had received a grant from the NIH that was a 7-year grant. It was for doing clinical research without any specifics about what the research was supposed to be. And therefore, the director, at his discretion, didn't have to get initially approval for the particular project he was doing. Well, the grant after that was for 5 years, and I think the one after that was for 5 years, and then they cut back to 3 years. But it, in effect, gave us an adequate amount of money. And in fact, on some years, I turned money back to the NIH because I didn't think we could use it well. And further, there weren't any, it was a division of clinical research, it was called, and there weren't any disciplinary restrictions. So we were able to do laboratory work, which was, I'd say, 70–80% of our time and effort. Clinical work, 4 or 5 of the members of the team were medically qualified, were MD clinical researchers. And so we did clinical, and we also were able to do field work, since we in effect had adequate amount of money, and we didn't have to argue about every time we did a field trip. It went very smoothly. So that combination of working in the laboratory, in the field, and in the clinic was feasible under the terms of the funds that Tom took the seat right here. We've saved for you. So, and it was in retrospect, it was a unusual arrangement, which actually doesn't occur very often. And I understand, I spoke to some people who awarded the Nobel Prize, and which was given at the same time to Carlton Gajicek, who did research very similar to that, field work, great deal of anthropology in his work. And he was a virologist by training and a pediatrician. And the two prizes, which were, he worked on Kuru and did some really spectacular work. But again, it was a combination of field work, clinical work, and laboratory work within the same organization. And that was cited by the Nobel Committee because you could start studying not only the virus, but its host, the variations in the host, populations in which the virus spread and caused disease, and also didn't cause disease, and then the behaviors of individuals and the behaviors in the population itself, all of which were really essential to our arriving at a very practical solution. So this started off as a basic project in clinical research. We did not start off, it was not a goal-directed project. That is, we didn't start off to find the hepatitis B virus. We were studying a phenomenon in nature with the expectation that if you do that and understand basic aspects of nature, that you will, in time, identify practical applications. And since many of us were clinically trained, the applications were quite apparent, and we didn't require any great urging to change the research from a kind of open-ended looking for new discoveries to a very directed project to see that the information was utilized. Now, I've been told that the best way to give a talk is to tell everybody what you're going to say, then say it, and then tell them what you said, on the expectation that they'll retain some of what you had to put across. So there'll be a certain amount of repetition. So this, I will make some further remarks about the process of discovery, but the take-home message has to do with the practical application of the research and the I would say, likely extension that what we've learned about hepatitis B virus and its prevention, primary, secondary prevention, possibly treatment. It's now the case for at least one other virus, for papillomavirus and probably hepatitis C virus. And it's going to be the case for many other cancers in the future, I think it's a prediction that emerges from what happened in this example. So to begin with, there's a hepatitis B virus is globally distributed. There are about 350 million to 400 million carriers in the world. There's about 40 million carriers of the AIDS virus, so it's about 10 times more common in that sense. And it's particularly common in Asia. Most of the hepatocellular carcinoma, HCC, primary cancer of the liver, in the US is due to HBV and HCV. Worldwide, most of the HCC, hepatocellular carcinoma, is due to hepatitis B virus. Now, it was identified here, the virus, hepatitis B virus, was identified in 1967. And as I already said, it was not the product of a goal-directed research. And I think it's important to keep this in mind, because the spirit of this institution has been basic discovery-based research. Research based on discovery of things you don't know, not finding things you do know. Now, it started off as an interest in inherited differences between people, specifically on the serum proteins, that were related to susceptibility to disease. Now, that is what we, at least a part of what's now referred to as personal medicine. Was the study of polymorphisms and how they relate to disease and how they relate to the environment. And it's what's now called SNPs more often than polymorphisms. And again, this was an open-ended project. It was an inductive study. That is, you went out into the field or the laboratory in many cases to see what was happening. And after you collected the data, you make the hypothesis. Now, there's no such thing as pure, that's inductive research. There's no such thing as pure induction. You always have a sort of general idea of why you're going to a particular place and why you're making particular measurements. But the notion is, broadly speaking, you collect the data first, then you make the hypotheses. The deductive part is you make the hypotheses very often based on your own data collection. That's always the most interesting. Then you collect data to test it. It's very difficult to fund inductive research. It's pejoratively referred to as a fishing expedition, which is a very strange metaphor, because if you want to catch fish, you should go on a fishing expedition. That is, if you want to get new ideas, there's no point to sitting around home and looking at what somebody else has done. Now, the applications commenced soon after discovery, and I'll have a chart showing that. We pretty quickly after the discovery of the virus, its practical application in testing blood donors to see if they were hepatitis carriers. That came very quickly, and by, I think we dated to about 1969, just 2 years after the discovery of the virus. The use of donor screening was started here in Philadelphia at what was Philadelphia General Hospital, but that no longer exists. And it was spread worldwide within a very short time. It's really quite amazing. Post-transfusion hepatitis due to hepatitis B had a very high occurrence rate because a lot of heroic, quotation mark, heroic surgery began after the Second World War, where patients would receive 8 or 10 units of blood, many of which were from hepatitis B carriers. And, but this allowed the detection of those, and within a few years after the institutionalization of the test, post-transfusion due to hepatitis B virus was pretty much over. And then the association of hepatitis B virus was with cancer was the first publication was with Dr. Smith and others from our lab that posited that the hepatitis B virus was one of the causes, one of the etiologies of primary cancer of the liver. And again, I'll come back to these points later in my effort to try to get you to remember what I had to say. Okay, so we're going to talk about primary prevention, secondary prevention, and the possibility of treatment of primary cancer of the liver and chronic hepatitis and acute hepatitis for that matter. So primary prevention obviously means you prevent a person from getting infected. So the vaccine was patented by Fox Chase Cancer Center in 1969. It took a while to get anybody interested in it. I mean, it wasn't much noticed because the whole discovery of the virus had occurred just a short time before that. But eventually we licensed a local company, one of the largest vaccine manufacturers in the United States, one of the few, by the way, and that's the Merck and Company was licensed by Fox Chase Cancer Center to develop and produce and market the drug. By 1980s, that had been tested by the late Dr. Wolf Shamunis in New York City. Based on his single trial, the vaccine was approved by the FDA and then later other organizations, and it became widely used beginning in the 1980s. And there's been a dramatic drop in the incidence of hepatitis B virus infection and the prevalence of HBV carriers, the incidence and the prevalence. And the prevalence or the incidence of hepatocellular carcinoma in the impacted populations, that is ones that had the vaccine, has dropped dramatically. Now, secondary prevention has to deal with people who are already infected with hepatitis B virus. And there are 350 to 400, possibly 400 million people worldwide who are currently carriers of hepatitis B virus. A significant portion of those people are at high risk of developing chronic liver disease, which is life-shortening, and primary cancer of the liver. Primary cancer of the liver is one of the most difficult cancers to treat. The 5-year survival rate is of the order of cancer of the pancreas, 5, 6, 7, 8%. 10% overall, and in some populations it goes down to 5 or 6%, 5-year survival rate. So treatment does not. Current treatment doesn't have a big impact on outcome, unfortunately. So, but you can treat people who are carriers, and and the big issue in clinical hepatology now is who you treat and when you start treating them. But there is growing evidence, I would say, that treatment of carriers greatly decreases the risk of developing primary cancer of the liver. And much of that was dependent on the work that was done in the last, 15 years or so by Tom London and his colleagues in Senegal and in China, which laid the groundwork for our understanding of the value of treatment. I'll show some of the data on this later. Now and I'll go into this and have more to say about it later. But again, this is in the way of an introduction, and there, if I have time, I'm going to talk about some of the local programs. Now, about a third to a half of the world's population has been infected with hepatitis B virus. Now, most of the people who are infected develop antibody against the surface antigen of the virus, and they have lifelong protection. A percentage of them develop acute hepatitis, and I'll show this again, and some of them go on to develop chronic infection with the hepatitis B virus, and they're the ones who are at increased risk of getting cancer of the liver and chronic liver disease. But stop and think, the world has had a very long interaction with this virus because there are viruses that are quite similar to this that have homologies, DNA homologies of 70 or 80% that exist in species that were evolutionarily prior to our own. And it's likely that the virus existed for a very long time, and the date's been recently established based on evidence in fossils, essentially. So it's been with us. We've interacted with this virus in a big way. A third, a half of the world's population has currently been exposed, and we've done it for a long time. That means there is an accommodation. There has been an accommodation, and based on polymorphism theory or evolutionary theory, that implies that there is some population advantages of having this, or having had this virus in the population. It'd be interesting to consider the consequences of that as the possibility of eradication arises. So about 25 to 40% die of cirrhosis, and these 300 million or so are currently infected. Now, that's our team in 1980. We've changed somewhat. And I think that was after we had a site visit. Wasn't that, Tom? I think that was the picture. So that was probably the maximum size of the group. And it includes our nurses and our sort of minister of secretarial staff and clinical research fellows, clinicians looking after the patients, and people managing the data and so forth. But it was quite a coherent group. We kept together for well 25 years or more. But when I was here, and then afterwards when Tom London did that.
The door didn't change much.
Sorry?
The door didn't change much. It's the same.
Yeah, that's right. That's probably one of the oldest parts of the whole, the whole complex. That building, of course, this building we're in. Okay, just to review some of what I've already said. When people are infected, most of them are asymptomatic. Some will resolve the infection, others go on to chronic infection. Some may develop acute hepatitis, but most of the people with acute hepatitis recover, but a small percentage will develop chronic infection. And then some, fortunately a small number, die of a fulminant hepatitis, but very difficult to treat. Some go on to cirrhosis, and then continue, and we may decompensate and die, and then some go on to hepatocellular carcinoma. Very difficult to actually state this figure, but it's probably in the order of 15–20%, depending on what the location is. It's very important to keep in mind that hepatitis B is one of the factors involved in the pathogenesis of primary cancer of the liver. There are other viruses that can be involved, like hepatitis C virus, and conceivably others. And in addition, there are other factors that interact with hepatitis B virus to lead to the outcome of having the cancer. And it's important to remember the fact every disease, to emphasize that fact, every disease has multiple factors. We're not so much interested in what you might call the real cause. We're interested in causes that you can do something about, that you can either prevent or that are targets. So that's one of the reasons we want to make a study of etiology as complex as possible. Because the more complex it is, the more things you can do, the more places you can intervene. Now this gives some idea of the prevalence in China, which is extremely high, and we're about 120 million, 125 million. United States, the estimate now is there's about 2 million carriers, and the EU slightly higher in southern and eastern, and Japan, which is about, was, because the epidemic has more or less been controlled in Japan, but it was several percent, rather higher than in the United States. And this was the initial finding that led to the discovery of the hepatitis B virus. So I'll spend just a few words repeating some of what I said before. Well, we were looking for inherited variation in the serum proteins, and we used electrophoretic techniques initially because in 1955, the gel method of electrophoresis was introduced. That's the one on which all molecular biology is based. That's the major technique. All those boring gels that you see use that gel. And you won't see any here, by the way. It'll be a very unusual presentation from that point of view, because much of this preceded the availability of molecular biology. In fact, all the major discoveries and practical applications were done before we had the possibility of doing, knowing about the genome. In any case, so then we used the technique that my colleague Anthony Allison recommended, and that is we had a hypothesis that if there are multiple variations in certain proteins, then people who receive a lot of transfusions are likely to receive proteins in the blood of the donors that they themselves have not inherited or acquired. And if these polymorphisms are antigenic, then the recipient, the patient, will develop antibodies. And we use this very simple, very insensitive method of double diffusion in agar gel, in which the antibody, that is the serum of a transfused patient, was placed here, and then surrounding it, we had wells in which you could put serum from a patient, from a person, from any individual, to see if they had a variant that interacted, a protein variant, an antigenic variant that interacted with the antibody that developed in the transfused patient. Well, Tony Allison and I actually found a polymorphism that that was detected in that way. We called it the AG system. They were antigenic variation on the low-density lipoproteins. And there were several years, there was a great deal of study of that, but again, at the proteomic level, which made the identification of variants extremely time-consuming and long. And that has proven to be very interesting. One of the first of the kind of modern connections of SNPs or polymorphisms with disease was between a variant of this lipoprotein polymorphism and Alzheimer's disease. And that's still one of the major pieces of information we have about genetic susceptibility to Alzheimer's. But then we continued the study, and we identified a variant, a reaction that was different than the first one that we found. And the original studies were done on serum from an Australian person that had been sent. We'd received that from colleagues in Australia. And we called it Australia antigen because we didn't know what it was. Now, when you name something that you've just found, you have to be very careful not to assign if you don't know a lot about it, you don't want to assign a functional name to it. That is, if you find something and you name it after the first disease you find it in, there's a kind of tacit assumption that it's connected with that disease, and it bars any other kinds of searching because you're sort of committed to a particular function. So to give something a geographic name means you don't make any, it's a proper name, like, like George or Henry or something, or Jane. That indicates that you don't know what it is, but you have to call it something, and that leaves your mind free, and you're not focused. You don't have your ego wrapped up in a particular function. So it's kind of it gives you more freedom. Okay, so then, as I said, we discovered this, and by 1916 we found this, and it was years of work on it, but by we first recognized the antigen as part of the hepatitis B virus in 1967. And that was by a combination of systematic studies, which I won't go into in detail here, which consisted of a series of observations, generation of hypotheses, doing the experiments, the designing and determining what the experiments you required, and then looking at the results, and then decide whether the hypothesis was supported or rejected. And then you made new hypotheses from the new data, because in testing the old hypothesis, you generate new data. This is a rather tedious way of going about things, but I think a very, first of all, it requires a real discipline. I'm now trying to acquire the discipline to write up in detail that process that we went through, and it's been at least 10 years that I started working on that. But in case, so there were applications that came quite quickly. The testing of blood donors actually started at Philadelphia General Hospital. Of course, we had a collaboration with them. In '69, Dr. Millman and I submitted a patent that belonged to the Fox Chase Cancer Center for a novel method for making a vaccine, which I'll tell you about. There was this paper that Bruce Smith was involved with that postulated HB was the cause of primary liver cancer. There had been intimations of that prior to our work. And then by '76, people more or less agreed that we'd found the virus. And in 1980s, the test was done. A very important piece of research was a prospective epidemiologic study done in Taiwan by Palmer Beasley and his Taiwanese colleagues that showed that if you looked at people who were carriers of hepatitis B virus but with no symptoms at the time of the study, and then compared them to others who were not carriers, and then looked forward prospectively, that there was, in his case, a 200-fold increased risk of getting primary cancer of the liver if you were a carrier of hepatitis B virus. Tom and I wrote a paper about important epidemiologic studies in the hepatitis B field, and this study really made was a very important, very well designed and conclusive, and that more or less convinced people that hepatitis B was one of the part of the pathogenesis. And I've already referred to the work that was done by Wolf Schmunis in New York City, which was it was accomplished with about 1,000 people in the vaccine trial. You know, half got the vaccine, half didn't. And the results were convincing, so much so that it was immediately approved. I should point out that using 1,000 people for testing, field testing a vaccine is unusual. 1.3 million children were part of the vaccine trial for polio. And I think one of the recent, sadly unsuccessful tests for an HIV vaccine was 20,000. It was a huge expenditure. And Wolf accomplished this because of the excellent design with a very small number. Now that's Manfred Bay is here. Manfred took the first images of the hepatitis B virus in our electron microscope group. This, I think, is a later image, not the first one. And in it, we made some observations that allowed going forward to make the vaccine in a rather unusual way. Well, the electron microscope, and by the way, this is a reproduction of the electron microscope picture on my tie. The glaring colors are not real colors, as we say in NASA. Okay, so so there's the whole virus, which is, at the time, it was discovered as one of the smallest viruses known. The hepatitis D virus is even smaller. It has a very small genome. It's very kind of intact. In addition to the surface antigen, there's a core antigen and the DNA and the genomic material about which a great deal is now known that's contained in the center. It was the first human virus that was sequenced. And that's because it's got a very small sequence, a small number of base pairs. And there's been an enormous amount of study, much of it done here at Fox Chase, to understand this virus. But an interesting feature that, by the way, none of us working on this project were virologists. Irv Millman had done his degree in microbiology, but it was primarily on bacteria, on tuberculosis. And the rest of us knew about hepatitis really from a clinical point of view. But we had to learn a lot about virology. I literally took the book on virology off the shelf to find out what a definition of a virus was and how you identify such. Okay, so in addition to this whole virus particle, there are these small particles and these elongated ones, which has the same diameter, which it turns out, and those are the ones we first identified under the microscope here. They appear to be all surface antigen. They have some differences from the surface antigen here, but we postulated that these particles would be antigenic and produce a protective antibody. Now, one of the reasons we thought that would be the case is that, number one, the only antibody we knew at the time was this one. And secondly, we had epidemiologic evidence, rather crude, I think you have to say, that it was likely that the antibody against this surface antigen would be protective. And that was based on the fact that we rarely saw, in fact, early on, never saw, a person who had both the antigen, this stuff and the stuff on the exterior, and the antibody against the antigen in his blood at the same time. Actually, it was a big advantage to using this insensitive gel immunodiffusion technique, because when we used a much more sensitive method that we also developed here, the radioactive method, we did find them both together. But there's a big advantage in science. You're looking for patterns, and sometimes you don't want the most sensitive method in order to discern patterns. That may be required later. But you want something that kind of delineates differences in an obvious way. Well, in any case, so a vaccine was devised that meant taking these particles, separating from the whole virus particles, discarding the virus particles, treating the remaining stuff with harsh methods to kill hepatitis B virus, for that matter, any known virus, and then adding preservatives and adjuvants in some cases. That was the vaccine. Now, it's the first time a vaccine had ever been made from the blood of a person who was a carrier of the virus. No one's made a vaccine like that since then, and I doubt very much that they will, because now there's a possibility of using recombinant methods, which would obviate some of the problems of using donor bloods. However, this was the vaccine that was tested by Dr. Shmunis. It was used on tens of millions of people, and as far as we can tell, there were no side effects from it. Nevertheless, people felt uncomfortable about using it, and as a consequence, when recombinant vaccine became available, that was widely used. But actually, the blood-derived vaccine was used up until quite recently, may still be in some places. The recombinant vaccine was made by several companies, and it was the first recombinant vaccine ever made, and there wasn't another one for, I don't know, 20 years or so. There aren't a lot of recombinant vaccine. Turns out it may not be the simplest way to go about it. I think I've recovered some of this. And this is a dramatic chart showing the distribution of carriers of hepatitis B virus. There are many of them in East Asia. That's probably the highest correction. In Oceania, in Africa, particularly sub-Saharan Africa. There's higher prevalences in Eastern and Southern Europe than in Northern Europe. And it's found widely in the Amazon basin, and also strangely in Inuits and Eskimos in the north. Eskimos are susceptible to infections. They live in a very closed environment for a good part of the time. And so within months of our identifying the virus, we knew a lot about its distribution because we had saved all the bloods that we'd collected when we were doing these studies on the distribution of polymorphisms in many parts. We'd collected bloods in most of these places that you see here, and many. So that's the distribution. Again, liver cancer, very low survival rates. It usually develops in the later decades of life, although most people in the world who get infected with hepatitis B virus are infected at the moment of birth or shortly thereafterwards, or conceivably or probably before birth. And they may remain asymptomatic for 20, 30, or 40 years and then develop the cancer, very often detectable when it's still small, and we'll get to that in a moment. And it's a very common cancer. It's not so common in the United States. By the way, it's one of the few cancers that's increasing in incidence in the United States now, probably due to the increased immigration of people from places where there's a high endemicity, Asia and Africa to some extent. And presumably that'll peak as more and more control becomes available and decrease. It's in places like Taiwan, it's the second most common cause of death in certain decades of males. And it's, I think overall it's estimated to be the fourth, fifth most common cancer in the world. Particularly, it's much more common in males than females. So it's a major contributor to the burden of cancer. Okay, so let's see what happened. The vaccination program starts in, let's say, 1982. It takes a little while for it to get going. Taiwan was one of the first places there was a national program. The US program went forward pretty well, but it took a few years for it to happen. It required legislation and so forth. And this is the incidence data for acute hepatitis, which is a reportable disease. Well, after the vaccine was licensed, it peaked. That's probably due to the fact that people were conscious of hepatitis B. They used the detection tests much more commonly, and so the reported number of cases probably went up as a consequence of that. But then it went down very rapidly when these procedures were instituted. The recommendation for immunization and recommended now in many jurisdictions it's required. So many of you who have young children, they will have had the hepatitis B vaccine. The goal is to deliver the vaccine to the newborn within 24 hours of birth. So they're often vaccinated on the table, as they're delivered before they can get away, so to speak. So, and now the vaccination program is worldwide. Something like 70 or 80% of the nations in the World Health Organization have national programs. So literally billions of doses of this stuff have been given. It requires 3 injections. And here's some other data that I've assembled from papers where it's possible to see what the prevalence of the carriers were before vaccination programs and afterwards. So these are, I think, useful way to put the data together. So, and for example, there's a regional study in China some years ago and dropped from 16% to 1.4%. In Hawaii, which has a high prevalence of cancer of the liver, by the way, it dropped from 1.6% to 0.04%. In places like Saudi Arabia, it dropped to very low levels, and indeed in Italy also. And by the way, in Italy and several other places, the prevalence dropped in the people who hadn't been vaccinated as well, probably because of a herd effect. You know, when the fewer people are infected, the chances of becoming infected decrease rapidly. So there was a long-term study here in the Gambia, again, a dramatic drop. And this has been the case worldwide. Most countries now use it, the vaccine. And I won't go into this. This was a big study that was done in China, and I thought which it was a sample of the whole country. It was a very well-designed epidemiologic study that was reported a couple of years ago. And I'll just sort of show some highlights from it. At the time, 20 million people suffered from chronic hepatitis B, and 300,000 died. This is in People's Republic of China. And the consequences, they showed that the reduction in prevalence was strongly associated with vaccination among all age groups. And they reached their goal of getting the prevalence in children down to less than 1%. That was actually, they went beyond their expected goal. And they did a kind of estimate here of the numbers of carriers that had been prevented. And that was, they estimated 16 to 20 million, and about 3 million deaths in China. Now you can make a kind of rough estimate to the whole world from that, and let's say there have been 29 million deaths prevented by vaccination up until about now. So that is, again, one of the major, one of the vaccines that's used most widely. Okay, now, then there was also data from Taiwan and from Korea in a reduction in the prevalence and the incidence of primary cancer of the liver. It dropped by about half. And there were similar reductions in Korea, rather more complicated study. And there's been a follow-up on this, and I'll just show this paper to say that the study that was done in Taiwan was repeated some 5 years afterwards, and it showed a continued drop, that the vaccine was effective in dropping primary cancer of the liver. So the World Health Organization 7, 8, 9 years ago categorized the HB vaccine as the first cancer prevention vaccine, with the expectation there'll be more. And there is another one that was introduced about 3 years ago, the vaccine against papillomavirus, which the preliminary studies are very convincing, and that now is becoming more and more widely used. Currently, it's far too expensive to use it for general population use. The story of the drop in hepatitis B vaccine is interesting. When it was first marketed, it was $100 a pop. And eventually, by competition and having other companies involved, it's less than $1. I think much less than $1 now. But that took years to happen. Now, this is a slide that was actually prepared by a drug company that's interested in this, in these data. It's in effect just a kind of market study, in which in different countries, the undiagnosed estimate, then those that are diagnosed and actually getting treatment, and those that are diagnosed and not being treated. So the US is probably as good as any place, and there's only 75% of them have not been diagnosed, 5% are treated, and 20 that are diagnosed, 20% are not treated. So there presumably is a big need for treatment. Now, one of the studies that was involved the Fox Chase group, this arduous work that was done in Senegal and in China, it showed that the probability, the risk of getting cancer is much higher when you have high levels of hepatitis B virus DNA detectable in the blood. than it is if you don't have any DNA detectable or if you have lower levels. Okay, so the argument from that is you want to decrease the level. And the way to do that is to treat the people who are carriers. And let's look at some further information on that. This was a prospective study with the same conclusion. The higher the level of the titers of the HBV, the greater the probability of getting cancer and chronic liver disease. And this is a similar chart for cirrhosis. That's for chronic liver disease. The other one was for primary cancer of the liver. So lower titers, lower risk. And this is, as far as I can tell, but perhaps someone will correct me, is the only study, a prospective therapeutic study, although I believe others are in progress. There were others published using drugs that are no longer widely used or no longer preferred, which showed the same thing for interferon, for welfiron, which has a similar conclusion. And that is that the placebos have had 21% disease progression compared to 5% for people who received the treatment. And there's a mutation in the hepatitis B virus that increases resistance to this drug. So lamivudine was the drug used here. And so those actually had an improvement as well, even though resistance developed. But what's required is a drug that is effective and has low resistance. And there are such drugs. There are at least 2 or maybe 3 of those now, which has very low levels of resistance and is probably the preferred drug for this. Okay, so the conclusion, if this is supported by further research, is that if you treat the drugs, you're going to get better. If you treat the carriers, you're going to develop, you're going to decrease risk. Well, there's a lot of issues. When do you start treating people? People get infected when they're born, essentially, or shortly afterwards. And the idea of starting somebody on an antiviral when they're very young, doing it for the next 50 years, we'd like to find a better solution. And there probably are better solutions. Now, one of them has to do with who's likely to get primary cancer of the liver among the carriers. And there are various factors you have to look at. First of all, things like, and that means you want to look at other factors that have an influence on you getting cancer. Well, strangely, this research, as I mentioned at the start, began as a study of the genetics of certain proteins, and our initial studies was on the genetics of hepatitis B virus, on susceptibility to hepatitis B virus. And there are now 8 or 10 polymorphic loci known that are related to susceptibility to becoming chronically infected with hepatitis B virus. So at multiple loci, if you have one allele and you're infected, you're likely to become a carrier. If you have another allele, an alternate allele, you're likely to develop antibody and therefore protected. There's also multiple loci that are related to the probability of getting cancer. So there are genetic factors that are involved. And understanding those factors is important. Now, there are also other agents that contribute to it. Alcohol probably contributes to the progression. There's been studies on certain toxins that infect cereals that aren't stored properly. Iron levels, certain protein iron storage levels are related to development of cancer. Things like arsenic are probably related. So there are a host of other agents that contribute to pathogenesis, and the more we know about them, the better we're able to treat the carriers and to prevent the progression onto cancer. So again, I want to emphasize the importance of understanding the virus, and we know so much more about that now because the genomics, or the genome, is better understood. Do you want to know about behaviors? A lot of the transmission of hepatitis B virus is due to things like behaviors such as tattooing. And by the way, one of the reasons for the recent popularity of tattooing was that it's now far safer than it was before the discovery of the hepatitis B virus. You know, shortly after it was clear that you could transmit hepatitis B virus by needle injection, illegal drug transmission is a major method of transmission. Well, shortly afterwards, it was clear that if you don't replace the needles when you're having tattooing, you have a real high risk of getting a hepatitis B and now hepatitis C. Well, laws were passed that required using replaceable needles, and that has led to a flowering of tattooing in our environment. You know, so we actually altered behavior, so to speak, by popularizing tattooing due to the, well, you get the story. And then there are other behaviors you want to control. You know, transmission from between children who are infected is quite common. Transmission, as I said, by injection of drugs is a method of transmission. So these are all behaviors that are related to it. And then there are things like cultural practices. And again, we want to understand as much as that as you can in order to effect control. Now, I'm just going to stop about now and say there are what you might call popular programs to control hepatitis B, and here in San Francisco is a kind of leader, and we have the Delaware Valley Liver Cancer Prevention Center, who was run by Tom Lennon and his colleagues for many years, and we got a very low-cost vaccine or free vaccine, and vaccinated hundreds of young children after birth. And before the vaccine was widely available. It was a very successful program. And there's now the Hepatitis B Foundation has a program in Philadelphia to hopefully make it a hepatitis B free place. And I will conclude and say that this is an important approach, and I've made a proposal to John when he's had a group of people sitting and talking to him, that we start a program here in which we are, I wouldn't say it would be a virus cancer program, but initially the approach would be to look at all the cancers that are known to be caused by viruses, and that is estimated to be 15 to 20%, and look for prevention, primary, secondary, and possibly treatment with antivirals. And then, as I've said multiple times now, diseases have multiple causes, and viruses are probably involved with the pathogenesis of many other cancers, have some role. So searching cancers to see if they have viruses in them using sophisticated methods, I think would be a very profitable approach. And setting up a program to do that means you can't help but find out something that's important. You could find out if viruses are involved, or during the course of the work, you'd find out other agents that are involved. So I'm suggesting that we have a program of that kind here. I've made this suggestion at multiple other places, and I think it's I'm very, how should I put it, sanguine about proposing at my kind of home institution. Well, thank you for listening. All right.
Can you tell us a little bit more about the genes whose genetic polymorphisms play a role? Are they all like MHC genes, or what are some of the genes that play a role in common?
I'm glad you asked, Maureen, because I'd like to show you the table if I can find it. It's there are multiple loci. Some the mannose protein. These are mostly that were identified very often with pre-genomic analysis. Rather than sort of guess them, because I haven't looked at them, but I can tell you there are multiple ones. Most of them you would not have expected. Well, the MHCs are very much involved. That's correct. But there are others that you would not. But It's a very tricky kind of study because what you do, you take a population, you look at alleles segregated into locus, and you see if the people who get cancer, let's say, or who get infected, who get chronically infected, if they have a higher prevalence of one allele compared to another. Well, it's hard to replicate that and find an association, and then you would expect to do it in another population and not find an association. That does not negate the first finding, but it tells you something else is going on in the other population. So, but those studies are quite feasible now. You can do them without getting out of your chair. You know, you can look at all the incredible amount of data that's available. I think.
Could animals living in your home, like a dog or birds, be imported from China or wherever, could they transmit?
Transmission from animals to humans has not been very studied. As a matter of fact, it's been very. We tried to get funding, to study the distribution of hepatitis B in animals, and if you look hard enough, you'll find it in a great deal of them. But there has not been, as far as I know, epidemiologic studies. By the way, we got very concerned about insect transmission, and we spent a lot of time in Africa and in Sarawak, wasn't it, where Walt Oxton went, or
Senegal.
Well, yeah, and then in Senegal, we collected a lot of mosquitoes. But that's tricky business, because if you collect these mosquitoes, you're likely to get bit by mosquitoes, and they carry a lot of bugs in them. And also, we looked at cockroaches. And cockroaches about 60, no, I take it back, bed bugs. We had one of the few tropical bed bug colonies in the world here at Fox. And the reason we studied bed bugs and not mosquitoes is if you go into an insectorium where you see a lot of mosquitoes flying around outside the cages. Bed bugs you can restrict because they, well, I won't go into the technology. But it's been, and I still think they may be potential transmitters, but there's no strong evidence that that's the case. Yes?
Barry, what's the biology? Why does hepatitis B cause liver cancer? Is it an insertional mutagen? Is it affecting is it more of an epigenetic phenomena? It has to do with the fact we're going to be sequencing all these cancers. If it's an insertional mutagen out of that project, we'll find out pretty quickly whether there are viral genomes that are in the DNA.
I think.
But if it's more of a, it's stimulating cell growth or something more indirect.
Well, the simple explanation is it kills cells, and you produce more. You have more possibility of second events or first
so
hits.
So that the tumor then itself wouldn't have hepatitis B genome in the tumor.
Yes. Oh no, hepatitis B is integrated.
It is integrated.
Oh, yeah.
So you would find it.
And transmitted from generation to generation, probably. It becomes part of our, that 7, 8, 9% of the genome that's viral.
So your prediction would be that if we were to sequence some primary hepatocellular carcinomas, we absolutely would find the virus genome there.
I wouldn't say, I think, John's the guy. John's now retired, but he may answer the question.
The woodchuck hepatitis is an insertional mutagen,
Yeah,
and that was discovered at the Philadelphia Zoo, and there's a number of genes that it inserts next to. With the human virus, it's rarely an insertional mutagen.
It did not follow.
It's integrated, but it's rarely insertion.
It's integrated, but not. One of the reasons it took a long time for people to say, well, this is a viral cause of cancer, where the epidemiologic evidence was very strong, is because it didn't fit a prior model. And the prior model came from the stuff that was done on mice and cows, and back in the 70s when the war on cancer started, and there was a big focus on cancer virology. But that stopped because the human cancers didn't follow that model.
Liver of the patients who carry the hepatitis B virus.
They very often do, and most people who get cancer have cirrhosis, fairly advanced cirrhosis, but not all. Sometimes they can go through. But yes, probably the major cause of death, or a major part, is the development, in some cases, of acute inflammatory reaction, acute hepatitis, and chronic hepatitis that doesn't go to cancer. I hope I've answered that. Well, thanks for staying so long.